Hello and welcome to a new episode of the Computomics podcast. Our guest today is one of Computomics' own, a technical account manager, Chris Kubica. Welcome.
Hi, good morning.
It's great to have you here. Chris, you may or may not know that we start with an icebreaker, and I was wondering, how did you even end up in plant bioinformatics? What was maybe one of the central reasons you got into that field?
Well, I guess it was kind of an accident. So first of all...
Those are the best.
Yes, it was a happy accident. I studied biology because one of my best friends studied biology at that time, and I thought it was cool. But I wasn't happy with all the lab work and standing in the lab for hours, so they offered this bioinformatics course, and I took it and I liked it, and so I ended up in plant bioinformatics. That's the whole story.
I like sometimes how it's these little decisions that really, they're like a door opener and you discover this new world, and it's great when it works that way.
Yeah.
And now you bring your expertise to Computomics. Maybe if we can firstly get into one of the themes or one of the topics you deal with every day is the topic of the pangenome. Why is it important for plant breeding? And maybe even what is it for those of our audience who may not know?
Okay, so let's start with what is a pangenome. In general, what we do in genomics is we have a reference genome. So that is the sequence DNA of mostly one individual that has been selected either based on specific traits it has to represent something that the first researcher that built it was interested in, or it was just one arbitrary individual that they had in their lab or from somewhere in the wild. And that already is a problem because this chosen individual does not contain, and it cannot contain, all variation of a species. So whenever we sequence something, so extract DNA from another individual, sequence it and compare it to the reference genome, we can only ever discover differences to the sequence that is present in this reference genome. So for example, if we take eye color, it's not a super good example, but if we assume that eye color is transferred by very different genes and the individual that we've chosen as the reference genome had, let's say, blue eye colors, but the one that we are interested in now has green eye color. The reference genome does not contain the gene for green eye color. So we can never, ever learn something about this gene because it's just not there to compare against. And using a pangenome, we can now limit and to a certain degree also overcome this problem because we're not relying anymore on a single reference genome, but we're taking a combination of multiple reference genomes combined into one reference structure, and that's what we call a pangenome. So we can represent more variation beyond what a single reference genome can do.
And why would you say is it especially important for plant breeding to work with the pangenome?
Plants are way more complex than anything else. Most research in bioinformatics is first being introduced in humans, and humans are very simple in their genetic architecture. That's just how it is. Plants are very complex, they are very repetitive, they have a lot of transposable elements. It's just a completely different game. And so they have a lot of different sequence that is not represented in the reference genome. For example, in maize, I'm not 100% sure on the numbers, but I think 30 to 40% of genes in the maize lines that are used for breeding are not represented in the reference genome.
That's a significant number.
Yes, and that's a problem if we're working with just reference genome. So if you're now using a pangenome, we potentially can't represent all 30 to 40%, but we can represent, let's say, 25 to 35%. You will still lack something, but you will have a way better understanding of your genetic potential using a pangenome than you can have using a linear reference genome.
So I would summarize it as you both get a more precise image and you also have more need for that because of the complexity of plant genomes.
Yes, and also because there's a lot of research has been done in plant genomes or in genomes in general. So everything or most of the things that could be discovered using the linear reference genome have been discovered and explained. So whatever is left, all the traits that are hard to nail down, they are potentially part of a region that is not really represented in the reference genome. So they can't be explained using the reference genome. So you need to move to a pangenome in order to get access to those traits and changes that cause them.
This is why we're talking to you specifically. At Computomics, you have something that SeedWorld dubbed the Google Maps of the pangenome, aka the app Pantograph. What is Pantograph?
Internally, we don't call it the Google Maps of Pantograph. We call it...
That's why I quoted SeedWorld.
I know, and I kind of like it, the name that they dubbed it. But so we call it an omics browser. Pantograph initially started out as a genome browser. So a way to visualize your pangenome. Normally in the bioinformatics field, there are tools like IGV, for example, where you can load your reference genome, your linear reference genome, and then scroll around, look at the genes, load your sequence alignments or regions of interest, and then just explore whatever is close to it. So close in the sense of in a genetic region next to it. That has not been possible with the pangenome because the pangenome is not linear anymore. You can't start upstream and end up somewhere downstream because there are connections, it can have loops, repetitive sequences, and so on. So there was a need to build a pangenome browser. And that's what Pantograph started as. And at some point, we figured out that just looking at your pangenome is not enough, and it's also not enough to charge money for. Yes, so what we need to add extra functionalities and values to just looking at the pangenome. So nowadays, you can use Pantograph to load all kinds of omics data, so variation, transcriptome data, orthology information, and so on. Everything that is being used in bioinformatics can be loaded into Pantograph, and then you can connect this information between the different samples that are part of your pangenome. So because the pangenome is not a linear reference anymore, but you have multiple references, and of course they are different, that will result in changes in, for example, their expression levels of genes. And if you have expression data from multiple sources, you can now load them into Pantograph and then compare them easily and explore them easily in a comparative way.
Can you describe, or maybe there's a way we can also include it in the show notes so people can get a sneak preview. How can we envision this visualization? What does it look like? How does it visualize the pangenome? As you said, there are many levels.
I guess the best way is to include a picture or to ask us for a demo, because we're always happy to give live demos to people that are interested in Pantograph and see it firsthand.
Oh, why not both? We'll add a little screenshot once that's been cleared. We'll add that to the show notes. And this is actually something I wanted to talk about as well, is we get to the demo maybe at the end again. We pick that point back up. But I was also wondering, how are your customers using the Pantograph? Because you just described it's a very powerful tool, but how exactly is it being used IRL, if you will?
Well, that's a very good question because there's no single way that customers use it. So I can shed light on two customers that we have that use it very, very differently. So the first customer, they actually use it as a genome browser. So they identify a QTL, a quantitative trait locus that they're interested in, and then they go to this location in Pantograph and check the structural variants. So the differences between all the samples that they have in their pangenome and overlap that with metadata that they have and SNP or variant data that they have and load it into Pantograph, and then try to pinpoint the specific change that causes this trait that they're interested in. And they were actually able to, very early on, identify a resistance trait that they have been hunting down for years.
Years?
Yes, years. And they couldn't figure it out. And within the first few months of using Pantograph, they actually identified the change and can now use it.
And was this because it was located in a way where it couldn't be found with traditional methods? Maybe if we can pick that point back up?
Yes, that was the case. So it was, I can't go into too many details here, but it was basically in a large structural variant that was hard to, pinpoint in the linear reference genome, and therefore now they could access it. So that's option A, using it as a pangenome browser, scrolling around, looking at things and exploring stuff. The second customer that is using it in a very, very different way is Pantograph, because it's a software tool, does not only have the front end that you can look at, so whatever is shown on your screen, but of course there's underlying software that automatically fetches information from a database or somewhere in storage to then display to the customer. But these endpoints can also be accessed by the customer themselves. So what they do is they automatically create reports based on, they have informations and positions that they're interested in, and they don't even really use the front end, but they take these positions in the genome, translate them into the positions in the pangenome, and then automatically create reports of whatever is in this region and more or less create a screenshot of the region to then give to their analysts.
So they use a different part of the app, mainly like an assistant for their own workflows.
Yes, they use it, I guess, more automated, more from a bioinformatics computer standpoint. So basically, Pantograph is a tool that can be used by bioinformaticians, which actually have a deep knowledge of computer engineering and so on, and also would normally perform all the bioinformatics tasks. But they can also be used by breeders, for example, that don't have a deep knowledge in bioinformatics, but are more used to the visualized way of exploring data.
Makes total sense. Can you, for this second example, or the client B, that is using it more in the kind of deep knowledge bioinformatics automated way, is there a similar story of how their workflows changed before and after Pantograph? Because it feels like something that would have quite a big impact on...
None that we know of yet.
Okay.
So no, I can't.
That would be interesting to explore maybe in a future episode. So let's talk about how this journey works. You mentioned you're happy to give demos to anyone who might be interested. So how would they go about contacting you for that? And then what's the process from there?
Once the initial contact has been established, what we normally do is we have, of course, a slide deck that shows what Pantograph is able to do just based on the slide deck. And then if customers are interested, they can get a live demo. And that means that we have a pretty nice example in soy where we can easily link a trait to a large structure variant and can then go through Pantograph and show the customer how you would basically pinpoint this change or this difference that you can see in the metadata of your different lines down to the actual change in the sequence that is causing that. And then also show them how to figure out which markers or how they can identify markers they can use to genotype this specific change in their lines. And then we use basically this example to go along all the functionalities of Pantograph. And if they're then still interested and we go further down the sales pipeline, what we would then do is go into a conversation with the client or potential client, what kind of data they have available and how that would fit into Pantograph.
I was going to ask about that because to me, that seems to be an important step because understanding the principle is one thing and the advantages are pretty almost self-explanatory. But then to think about, okay, but what specifically does a client need? And also what type of resources, what kind of data, what kind of omics data also could they even provide? I would assume that can also be a challenge or it's almost an educational step that's already helpful as well.
The most basic version of Pantograph needs a reference genome, and that's it. Of course, then it's not different from a normal genome browser because there's no pangenome involved. Your pangenome consists of one reference genome, that's it. But you can add as many reference genomes or genome assemblies to your one reference genome as you would like. And those can, of course, come from the public domain, which a lot of our clients do. And then they enrich them with their own proprietary data. So either reference genomes that they have sequenced internally based on their own lines, or genotyping data or variation data of their own lines that they then put into the pangenome context.
That's been cleared up. I'm assuming there will be some kind of agreement and then they're immediately ready to go, or what's the next step to go into basically the actual use of Pantograph?
Once we have agreed on all the data that they can provide us or that we collect for them, there's always a setup phase where we build their pangenome for them. And depending on the size of the pangenome, the species, the number of lines, yada, 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 that is weeks to maybe months in time. And once that is done, they get a license for Pantograph and then they're able to use it using the pangenome that we provided for them.
And do you provide assistance? Because beyond obviously the initial understanding, key questions, what type of data, what kind of assistance is part of the Pantograph license with you?
Apart from the setup phase, there are for sure 2 training sessions that we give each client. One is the initial handover of the graph where we show the client how to use Pantograph, how to execute all the functionalities with their own data and so on. And then the second training session already is more open, where we don't really do a training anymore, but we, based on the customer feedback and what they are most interested in, we go into more detail on the actual tasks that they want to execute using Pantograph. And then, of course, if anything comes up, any questions arise, we're always available for another short meeting to clear that up and figure out if there's something that we can do. And what we in addition do, we collect, wishes from our customers, what they would like to see as features in Pantograph. Because although all of the people that work on Pantograph have a bioinformatics background, in the background in biology or breeding, we don't use the tool, we just develop it. what is really helpful to us and really helpful to our clients is they give us ideas of the direction in which we would like to develop Pantograph. So based on customer feedback, we already implemented quite a few features of Pantograph that are now in use that we haven't thought of before. What you can do in Pantograph is you can export sequence. So you select a region in your view, and then you click export, and you get the sequence of whatever you selected. And what we never thought about is people, of course, would also like to get all the variation and the annotation and also the underlying alignment. So they can get the alignment of the region that is viewed by them, basically. And then we updated so Pantograph comes with an option to not only view the pangenome, but also use it, for example, for genotyping. So what you can do with Pantograph is you have sequencing data of some lines that you're interested in. And what you would normally do is you map them against reference genome and then call variants, or map it against the set of known variation and genotype it. What you can now do is you can map it against the pangenome and not only have access to the sequence in the reference genome, but to the sequence in the full pangenome. And we expanded and added new genotyping methods to our existing genotyping method based on customer feedback.
Well, that naturally leads to the question, what are current items on the wish list, or what can we expect? What is planned maybe for Pantograph in 2026? Let's keep it to this year.
So I guess one of the big things will be a BLAST functionality. So BLAST is a household tool of bioinformatics where you can...
as a non-bioinformatics person, I'm like, that sounds kind of dangerous and interesting. What does it mean?
We don't demolish or explode anything.
Shame.
Yeah, I know. But what you can do is you can easily or rapidly search for small snippets of sequence. So for example, you are interested, you know that a certain gene in tomato confers a resistance to something. And now you're interested in your maze lines. Do your maze lines contain this gene or a close relative to this gene? So normally what you would do is you would take the annotation of your reference genome, get all the protein sequences, and then blast this tomato protein against the proteins in your reference genome. But as we already talked about, maze, there are like 30 to 40% of the genes in the commercial lines, they are not present in the reference genome. So with a pangenome, or with BLAST against the pangenome, you can now not only blast against the sequences in the reference annotation, but against all sequences in the pangenome. And that will be possible for proteins, so the protein sequence, the coding sequence, which is the sequence before the RNA sequence, before it gets turned into a protein, and then also against the sequences of the assemblies themselves.
I assume that would also increase the likelihood that you will find interesting traits or interesting areas quicker.
Yes, that's the idea behind it.
Any other features that you're looking at? That already sounds like a pretty big one. Any other features you've got planned?
So something that might happen based on that, based on the BLAST functionality is, so currently what we do in Pantograph is we build our graphs based on chromosomes. Because sadly, as we all know, computational resources are limited, and with AI, they become more and more limited. So what we need to do in order to utilize the resources that we have available is we need to limit each pangenome graph to sequences corresponding to the same chromosome. So all sequences corresponding to chromosome 1 go into one graph, chromosome 2 into another graph, and so on. And then in Pantograph, you can visualize them all together. But nevertheless, you click on chromosome 1 and you see the graph for chromosome 1. And you can't see sequence that is linked to sequence on chromosome 2. So with the BLAST functionality, you can now blast sequences against the pangenome and identify sequences on chromosome 1 and 5 and 8 and 20. And what we envision now is to, after identifying sequences using BLAST, go back and perform another graph construction step on the fly to draw together sequences that are related on different chromosomes into one new subgraph, which have not been aligned before. And that will then give you the power to explore variation of sequences that, for example, are transposed from one chromosome to the other and have not been linked in Pantograph in a pangenome setting.
That's exciting stuff. Is there a way you're rolling this out to existing clients, or maybe a webinar or some type of informational output that we can look forward to?
Yes. In terms of rollouts, until now, all new developments that we do internally will get released to our clients. So when you have a Pantograph license, every time there's an update, every client gets that update. What we are planning to do this year is we are in the early stages of planning a webinar with a customer of us that will go into detail of how they use our technology and how they explore it and what value it added to their company.
How can people sign up for this, or who is it targeted for? Can we give our audience maybe a hint on how to participate if they're interested in it?
So it will be announced on our social media channels for sure. So especially on our LinkedIn, I guess. I'm not 100% sure, but it might happen together with SeedWorld. So then it would be announced on the SeedWorld page as well. On our website for sure. And I guess it's aimed at everyone that is interested in pangenomes and seeing what pangenomes can add to their tech stack, basically.
I get the sense from what you've shared in this episode today that there will be quite a few people who are interested in it if they aren't aware of it already. And with an eye to time, Chris, thank you so much for giving us that insight, all those exciting new things we can look forward to with Pantograph and what you've already been doing. I hope you will be back with more insights at some later point, either this season or next.
Well, I hope so too. Thanks for having me. It was a blast.
Nice one.
Thank you for listening in today. Want to learn more? Then check out our show notes and more info on Computomics.com. If you have questions or want to propose a next guest, please reach out to us at podcast@Computomics.com.